Hi, my name is Chamia Chatman. I am advised by Dr. Erica Majumder at the University of Wisconsin-Madison. Today, I'll be discussing my thesis work on the effect of a groundwater contaminant mixture on gastrointestinal cell viability. Just advance this way. So within the state of Wisconsin, agriculture is a major economic sector. With that comes the use of millions of pounds of fertilizer and pesticides to maintain crop production. Unfortunately, as a result of that usage, groundwater contamination due to agricultural runoff has become an increasing concern, specifically because this increases potential health effects to not only humans, but also livestock that are drinking this now contaminated groundwater. Therefore, our lab aimed to identify and evaluate if and how these agricultural mixtures detected in Wisconsin groundwater would affect CACO2 cell viability. The CACO2 cells are a gastrointestinal cell line derived from humans and are typically used for toxicology research. Within the study, I did hypothesize that exposure to a mixture of these agricultural chemicals would lead to greater decreased cell viability compared to exposing these cells to these chemicals individually. So within this study, I aimed to break this down into three components. The first component was first identifying measured concentration data taken from the 2021 Department of Agriculture, Trade, and Consumer Protection's targeted sampling report. And this report specifically looked at 104 samples from 16 counties and agricultural areas throughout the state of Wisconsin. In part two, I then utilized computational tools toxEval and toxmixtures, which are developed by the USGS, to identify chemicals and chemical mixtures within this concentration data set. And then part three was to perform my own biological assays on our new assumed chemical mixture of interest and look at two endpoints. First, an MTT assay, which is a colorimetric assay to evaluate this reduction of the MTT to formazine through the NADPH oxidoreductases, and it turns a purple color. And then also with a cell viability stain using a double stain of prapidium iodine, which is a membrane permeate dye, and then hoax, which binds to the nucleus. So moving into the results for part one, again, utilizing this 2021 DATCP report, I aim to look at whether or not there were chemicals that were frequently co-occurring together and were also above groundwater regulations, specifically an enforcement standard or preventative action limit. And by doing so, I was able to identify within 4 counties that nitrate, which is a naturally occurring chemical typically found from fertilizers or human and animal waste, was above these regulations, and also co-occurred with an insecticide called atrazine and a herbicide called atrazine and an insecticide, imidacloprid, excuse me. And by doing so, we then had chemicals of interest identified. Moving into the second part of this study, I'll take your attention to the top left table, which is our toxEval output. And this output was broken down into 2 parts. First, calculating exposure activity ratios for chemicals of interest to help us prioritize which chemicals would lead to biological impact, and then also correlating that to specific assays within the US EPA's ToxCast chemical screening database. So in this table, we were able to pinpoint 9 chemicals, 8 of which were herbicides that resulted in several active hit calls, as they refer to it as, or biological assays in which these particular chemicals resulted in a biological effect. So some of these endpoints included xenobiotic metabolism mostly. So this became very interesting for us because we noticed that atrazine was one of our initially screened chemicals of interest. Moving into the second computational tool utilized, the second table here is from our tox mixtures output. This was able to identify 1 groundwater well, which was DB719 in Waupaca County, which screened then 5 chemicals of interest, which led to these adverse health outcomes listed on the far right of this table, which included thyroid hormone catabolism, as well as increased activation of the acetylcholine receptor, as well as cycloogenase inhibition. I boxed in orange the 2 chemicals which were initially screened by us by our screening of the report. So that's the insecticide imidacloprid, as well as atrazine, the herbicide. So at this point, we did make the conscious decision to still focus on our initially screened chemicals, which included nitrate, atrazine, and imidacloprid for our biological assays. So again, just to reiterate, these biological assays included exposure to our 3 chemicals using the CACO2 cell line, again, having that double stain of propydium iodine and hoax, and then overlaying those 2 on the far right. Interestingly, we did not notice greater decreased cell viability with this particular mixture, which incorporated the exact values from the 2021 DATCP report. However, a prior experiment using checkerboard assays revealed that 10,000 parts per billion of atrazine and nitrate and imidacloprid, excuse me, but 1,000 parts per billion of nitrate actually did result in increased growth inhibition to chick and cecal samples. And utilizing those same concentrations in this cell culture model and experiment, we also saw greater decreased cell viability. And that was also seen with our MTT assays. So to conclude quickly, just emphasizing that within this model and this study, in silico methodologies really provided computational power to our ability to predict which compounds would be of highest concern. And it was very interesting that 2 of our 3 initially screened chemicals did appear within that output. And then lastly, just emphasizing the point that performing biological assays at environmentally relevant concentrations is vital for understanding human and animal impacts and health outcomes long-term. So thank you for your time.
Excellent, thank you, Chamia.